sequenced your plasmids? If not, stop what you're doing right now and go sequence them. Okay, maybe don't stop what you're doing right now, but you shouldn't be doing any work with a plasmid until you verified its sequence. So this could be something you just cloned, this could be something you ordered from a company, this could be something your lab mate gave you, but anytime you get a plasmid, you want to verify that the sequence is what you think it is, and even that the insert is what you think it is. You could have gotten a plasmid with a totally different gene inside. That happened to me once, so true story, it happens. Even if it's not a whole different gene, if there are typos in it, that could be messing up your subcloning from it, as well as expression from it, and all sorts of different things. Bottom line, you save a lot of time and energy in the long run if you verify that sequence in the beginning, or at least you can if there's a mistake. And so today I want to talk about some of the practical details of sequencing plasmids, as well as analyzing the sequencing data. So mostly talking about like Sanger sequencing and designing the primers for this and things, but also you can also do whole plasmid sequencing. So let's get into the details. So I'm just going to dive in. If you need more background, I have lots of other videos and posts that I will point you to. But so we're all on the same page. We have this plasmid, we have this circular piece of DNA that bacteria will make copies of for us and will pass down from bacteria to bacteria. And this plasmid is often serving as a vector or a vehicle for getting a piece of genetic information into these bacteria, for the bacteria to make copies of, to make proteins of, etc. And so these plasmids typically have this kind of unique part, which is going to be your insert, the gene or the thing that you put in there. And it's going to be be combined with this vector backbone. And so this vector backbone is going to be kind of generic. There are different ones. There's like pet vectors and puck vectors and various vectors. But different researchers will use the same vector backbone with different inserts. And it's often this insert region that's what we really care about when we're doing this sequencing. An alternative method is going to be whole plasmid sequencing, which is getting more affordable, and that'll basically allow you to sequence the entire plasmid. But we're going to focus today on the traditional sequencing. When we talk about sequencing, we're talking about this like Sanger sequencing, where we're basically going to design primers that are going to sequence this insert. And I'll tell you more about how we do this. But first, a little about why we're doing this. So basically, the plasmid in that generic part is going to have a selection marker, which is typically an antibacterial resistance gene. This will make it so that you can select for bacteria that only allow bacteria to grow if they actually have that antibiotic. So resistance product, so meaning that they have the plasmid. But just because they have the plasmid doesn't mean they have the insert. There are ways that we can test whether this plasmid probably has the insert, things like colony PCR and analytical restriction. But even these methods can't tell you whether the sequence is correct. And the only way to know that is to actually send it for sequencing. And when we're sending it for sequencing, we're typically talking about Sanger sequencing, which is the traditional method of sequencing, this dideoxy chain termination DNA sequencing. How it works is kind of like PCR, except that you only have one primer per reaction. And the copy is kind of you're going to use poisoned nucleotides, kind of. So deoxynucleotides are normal DNA letters, and so they get that deoxy because they don't have this 2' OH, which is what RNA have. But they still have a 3' OH, and this 3' OH is going to allow them to link together. Now, a dideoxynucleotide, basically, this is also missing this 2' OH, or this 3' OH. And if it doesn't have that 3' OH, well, then it can't link up to other DNA letters. And if it can't link up to other letters, well, now you're going to terminate a chain. And if you have a dideoxynucleotide that has a label on it, like a fluorescent label, now when you terminate the chain, you're also going to be labeling the DNA. And if you put in your reaction, mostly deoxynucleotides, so mostly ones that can link up, normal ones. But then also some of these labeled dideoxynucleotides, you'll basically get products of different lengths that are going to be labeled. And you can label each of the different DNA letters, so C, A, G, and T, with a different fluorophore or some other different label. And now your chains, these different length chains, are all going to end with one of these fluorophores. And by reading out the fluorescence, you are able to then read out the sequence of the DNA. And so this is the idea behind Sanger sequencing. And so like PCR, you need to design a primer in order to tell the DNA polymerase where to start. But unlike PCR, you're only going to have a single primer per reaction. You're not going to be making copies. You're not going to be making lots of copies over and over cycling. Instead, you're just going to basically have this single kind of cycle where you're not even making a full product each time. But the idea of the primers is going to be the same, where you're basically going to need to have primers that are going to tell polymerase where to copy. And often what we're doing is we're wanting it to copy into the insert. Now, thankfully, because plasmids typically have some sort of generic backbone, you can often kind of like buy, or even have the sequencing companies will put it in for you for free, often kind of general sequencing primers that bind to the generic parts of these vectors. So typically in the region around the insert, there's going to be like an MCS, or a multiple cloning site, which basically has restriction sites and things like this that you can clone into. And it's also going to have like a promoter and things like this, these basic features that are going to be right next to the insert, no matter what insert is in there. And so if you use primers that start outside of in kind of like going into your insert, but one per tube, remember, not both in the same tube, well, now you can basically get your insert sequenced without having to design custom primers in order to do so. But this might not be sufficient for you. Even if there are primers that will go into the beginning and the end of your insert, basically each of those Sanger sequencing reads is only going to be about 500 to 1,000 bases. And the first like 650 or so aren't going to be reliable. So the beginnings and the ends of the reads are going to be unreliable, as we'll look at. So when you design the primers, if you have a long insert, you might have to design an internal primer to make sure that the read from the forward primer and the read from the reverse primer and your internal primer, that together they kind of like overlap and give you the entire sequence. So whether it's a primer that's for the vector or a primer that's for internal, typically these primers should be about 20 base pairs and start about 60 base pairs before the region you want sequenced, because the beginning is going to be unreliable. And you can look and see on like sequencing facilities, they'll often have a list of universal primers or something, they'll call it something like that. Often, basically, these are going to be generic for the backbone, but they're different ones for different vectors. But often you can have them add that primer for you. Whereas if you want a custom primer, then typically you pre-mix it in with your vector when you send it for sequencing. And so typically what you're doing is you do some sort of mini prep, you purify out the plasmid, and then you put some of that, you measure the concentration, you dilute it to whatever amount that the company wants. Then you either mix in the primer that you want to use, or you tell them use this general primer that they already have, and they'll put it in for you. Then you like print out an order form and you stick it and you bubble wrap your sample up, and you go and you stick it in some sort of like sequencing pickup box or something like this. The details are going to vary from place to place, and depending on whether you're doing it like your institution is doing the sequencing, or you're sending it off to some company like GeneWiz or something like that. But then it's typically pretty fast. So a couple of days later, you're going to get back the results. And so how do you analyze these results? So the most important thing is that you actually look at the data. And so don't just look at this like computed sequence that they give you. They're going to send you a few different types of files. And so some of them are just going to be like the sequence, the actual sequence, at least what they say the sequence is. But that sequence that was kind of like called is going to be based on the actual data. And you can see the actual data in the trace files, so these .ab files, which you can visualize using software like CLC Main, Snapgene, or DNA Star. And also in that software, you're able to align them to your reference sequence. So what you think the sequence should be. So basically the trace you see here is going to be a chromatogram showing you like the different DNA letters, the traces from them, the fluorescent traces from those dye molecules on those different dideoxynucleotides. And so each of them, the C, the T, the A, the G, they each had a different fluorophore. And so you'll have 4 different channels, but they're going to be overlaid. And so you get this one kind of like overlapping trace where these different colors are going to be showing you which DNA letter was read at that position. And typically this is also going to be written above in the in like small letters, as well as often it'll show you the translated version. And so the protein letters that accompany the DNA letters, and then the DNA letters, they're being read out or called based on the trace down here. Now, the reading frame will vary depending, so the translation, it'll actually vary depending on your reading frame, and much more on that in other posts, as well as how you can kind of check out different reading frames. I'm not going to go into the details because it's going to vary from software to software, but there are typically ways where you can view the different reading frames if you're interested in that. But what we're interested here is going to be looking at these AB traces, looking at how they compare to the reference sequence, and looking at where we should what we should be looking at. So first of all, when we look at these traces, we want to see a nice clean trace like this. We don't want to see overlapping traces, which would indicate that we have like multiple sequences present. We have some sort of like contamination. There are multiple plasmids in there, which wouldn't be good. Basically, what you should have is you should have this nice clean signal. And if you do, you should get a consensus sequence. So when I was talking about these little letters up here telling you which DNA letter was read out, this is going to be the consensus sequence. So if you had multiple sequences in here, this is going to be the most prominent nucleotide at that position. But sometimes there's going to be multiple traces, and the software isn't going to be able to figure out what is actually there. And so you'll see something like an N or an X, which is basically saying that the software can't make the call because you have these overlapping traces. You can also get problems if you have it so that there's multiple of a letter in a row that it can kind of blend together. So if you had like multiple A's or multiple G's or multiple C's, you can kind of get these traces that are going to kind of overlap, or kind of you don't see the big dip in between them. And this can make it hard for the software to make the right call. And so the software will often tell you like where there's conflicts, or they'll just say that there's, they'll call it wrong. They'll make a choice. And this might not actually be the right choice, or they might put an N where you can go and look at it and you'll see, oh, well, that's clearly just because you have these 2 G's right next to each other. You can see that you actually do see these 2 separate peaks, and it is GG. So your eyes can tell more than the software can sometimes. And so if you were to just go and look at the consensus sequence, which is what that sequence file would tell you would be the consensus sequence, it would have those N's, or it would have those X's, or it would have those wrong letters. And so it's important that you actually go and you look at the data directly, go to those conflict sites and look and see, okay, well, can I figure this out? Now then if there is a conflict, you need to decide, well, does it really matter? So sometimes there's going to be a synonymous mutation where basically the DNA letters change, the DNA sequence changes, but the actual protein sequence doesn't change. And so maybe you don't care. And maybe it was actually a misannotation in your original reference. And so I've had this happen where there's a conflict with the reference sequence, but it's a synonymous mutation and it's present in all of the clones. And so all of the different copies, like if I tested out like multiple clones, and they all had the same mutation in there, which didn't change anything. And so it was probably just a misannotation of that original reference. But it would be important to know if I was going to design, say, a primer later that was actually going to cover that region. So in terms of the actual protein making, doesn't matter. In terms of if I was designing a primer that I needed to know the sequence, then it could matter. And sometimes you'll get mutations that really matter for both cases, that they're non-synonymous. So they change the protein letter that's spelled for. And so these are clones that you definitely want to get rid of, as long as that signal is reliable, however. And so if you have a signal where it's kind of like unreliable, well, then that's a different story. It might still be okay. And why might it be unreliable? Well, sometimes this happens in the beginning and the ends of the reads, in addition to those things like those spaces being close together or having contaminating things where the louder signal is kind of going to outcompete. In the beginnings and the ends, so basically like the first 50 and last 50 or so like nucleotides that are read out, you can't really trust them. And so often they'll have like ends and then. But sometimes they'll actually try to guess some of them and just basically don't trust this. And this is why when you're designing your primers, you want to make sure that you're designing your primers to start like 50 or 60 or so before the region that you actually want copied. Because you're going to have these weak, messy beginning and end. And so this might mean that you're going to have to design primers that are multiple primers just to get the whole insert. So if you do like a forward and a reverse on the different ends of the primers, like with those generic vector primers, if your gene isn't too long, if your insert isn't too long, then they'll overlap with one another and you get the whole insert covered. But if your insert is longer, then you're going to have to make an internal primer as well. And if there's a sequence that is like in the kind of unreliable zone, you definitely want to make it so that you have a primer that is going to be definitely cover that region of interest. So like start like 60 or so before that region that you really, really care about. So that is the basics of how to interpret this data. And so there's a couple places you want to look most closely are at like the beginning and the end, the places where you actually cloned in a sequence where the insert meets the vector. And then you're also going to want to look at any region that you tampered with. So if you were making a mutation to a region, you want to check and make sure, well, did that mutation really take? And then, of course, you want to look at all of the other places. But if nothing else, look at those sites. So that's typically what your eyes should go to first. That's with the place where there's most likely to be a problem. And remember, don't just look at the sequence file. This is going to tell you the consensus sequence. That can be fooled into thinking that the sequence is wrong when really it just called it wrong. Or maybe there was, you have contamination, which is going to be a bigger problem. Sometimes when you go to align the sequence, you find like, okay, well, this isn't actually aligning, especially if you just start with with the sequence file and then you try to align it. And so if you're trying to like kind of manually align it, you're going to want to choose a part of the sequence that's actually in the middle, because remember those ends are going to be unreliable. And so you can take like a middle part of the sequence and then try to align that and align the whole thing based on that. Sometimes the sequence, like, you're like, where the heck does this come from? It's not matching any place in my plasmid. So sometimes you might see a sequence and it's going to kind of match elsewhere in your plasmid, not where you were expecting. You might see just the plasmid, the vector, instead of seeing your insert in there, things like this. But sometimes you can't even figure out like where the heck this sequence came from. And so then you can do BLAST and kind of like look up the sequence, see where it came from. And sometimes you might find that it's something that your lab mate was working on, or it's just some really random thing that you have no clue how it got there. Sometimes what you can do is you can actually have the sequencing company like run it again, because sometimes there might be an error on their end where they give you someone else's results or something like that. But sometimes it really is that your plasmid is just has something random in there and who knows where it came from. But you wouldn't even know about that. You'd just be kind of helplessly trying to clone out of it or express from it if you didn't sequence it. So the first thing you do when you get a plasmid, the first thing you do when you clone a plasmid, the first time you do anything with a new plasmid is you want to sequence it. And so often when you order a plasmid, it'll come as like an agar stab or something like this. You can grow it up, make a mini prep of it, and then send it for sequencing before you try to do too much with it. So I hope this helped you, and best of luck with your cloning, with your expressing, with all of your work with the plasmid. Remember to sequence it before you do too much, really anything with it.